Hi, so my name is Hannah Nilsson, and I am a first year graduate student at the University of California, Berkeley, in the Molecular and Cellular Biology program. And I'm here today to talk a little bit about my science and how I got involved in it for Be a Scientist. And so before I get into that, I just wanted to kind of tell you a little bit about myself and what I like to do outside of science. I love to go outside and I really love dogs. This is my dog here. I also love to ski. I love traveling. I love listening to music and I like making really ugly baked goods. This is a cake for my roommate, and this is a fly cookie that is science inspired. And so, just to give you guys a little bit of flavor about who we are outside of the lab and that we're kind of normal as well. And just to give you a little bit of background about why I got into science, when I was in middle school, I saw this crazy video about just the scale of the universe. And it started by zooming out and showing just how big the universe was. And I thought that was interesting, but it didn't really impact me in any way. And then it kind of zoomed back in and went into how tiny things were really. And that got me really excited about just kind of what was around me and got me thinking. And so this is just kind of a sample of that. And what you'll see here is it starts with the scale of the universe. And so this is an unimaginably small number, but it's just, these are the things that make up our life. And so it's just showing you kind of how we zoom back in from there. And so I just thought this was really cool because I was interested in these things that I couldn't see, but that made up pretty much everything that I am actually able to see. And so this is just a little glimpse at how cool science is, and just how broad it is, how big things can be in the universe. And so I would encourage you to look up the rest of the video on your own time, because again, you might not be interested in the small things like I am, but it's also really cool to just see how far everything expands. And so just kind of building off of how excited I was about the small things, I got very interested in microscopy. And so there are several different types of microscopy, and each type of microscopy is able to tell you something that's different. And so what you'll see here is there's something called confocal microscopes, and those microscopes are the ones that you've probably seen in your classroom. They're what you can use to look at like plant cells and other organisms as a whole. And then there are other microscopes that are able to look at smaller things like the building blocks of cells, so proteins, DNA, sugars, and fats. And you can use fluorescence to kind of mark those things and look at those things in color. And then there's even something called an electron microscope, and electron microscopes can go even further down and show you in great detail how those building blocks are made up. So not only can you see a protein, but you can see all of the individual atoms that are in the protein. So I have fallen in love with all things microscopy and all the different things that you can see with microscopes. And so just a little foray into how I've been able to use microscopes is my first research experience was when I went to college. It was in Minnesota, so it was really cold there, but I got to do some research on these things called Thermophila, and you'll see them right here. I like this image because it was one of the first images that I was able to take on a microscope, but also they look like hearts. And Thermophila are unicellular organisms, which means they are a single cell, and they live in fresh water. So I can almost guarantee you that you all have swallowed tetrahymena at one point in your life. They can do some really cool things. They can reproduce asexually, which means that they just make little copies of themselves, or they can reproduce sexually, which means they make entirely new kind of versions of themselves. And what I was interested in is when they mate, they're able to fuse together and then they can come back out into separate individual cells afterwards. And so we wanted to understand, using microscopy, how those cells are able to come back apart after they've fused together. And so we were able to do this by looking very, very close down. So as I mentioned before, there are different types of microscopy, and I was able to use an electron microscope to look at where those two tetrahymena kiss each other, basically, and how they then come back apart. And we were able to look at that. Using that microscope, we were able to model what it was like, and then we were even able to 3D print what we were seeing under the microscope and interact with it in our hands. So it's, again, microscopy is super cool because we can take something that we can't even think about without that tool, and then we can model it in different colors, and we can actually hold it in our hands by using those models. And so it's not just about pond scum, though. I've been able to use microscopy to look at other things. I've used something called fluorescent microscopy, which is a technique that uses different kinds of stains to look at different molecules in cells. And so here, these are just human cells, and they've been stained for different proteins. And I was able to look at how those proteins interact with one another. And as you can see here, sometimes they don't interact, and that's when the colors stay separate. But sometimes those proteins actually do come together, and that's when you see these kind of yellow dots here. And so, again, it's not just about a single-celled organism living in a pond. Through microscopy, we're also able to understand cells in our own bodies and how they work. And then what I study now, so this is something called a scanning electron microscope image. And this is just an image of bacteria. And you can look at the surfaces of these bacteria, and you can almost visualize them in 3D space using some of these microscopy techniques. And what I'm interested in using microscopy for now is to understand how we humans and other animals and organisms are able to battle these pathogens. So things like bacteria or viruses or fungus, and kind of what the interplay between those two different beings are. And so what I'm interested in studying is Mycobacterium tuberculosis. So it's the bacteria that causes tuberculosis. And basically what we're trying to understand is how this bacteria is able to live within human cells, and how human cells are able to either combat them by using something called a lysosome to degrade the bacteria and break down its walls, or how the bacteria is able to kind of hijack the host cell to produce its own materials that it needs to survive. So just starting to pick apart and dissect the delicate interplay between those two kind of systems, and how we're able to fight bacteria efficiently, but also how bacteria are able to infect us. And so again, we want to use microscopy to start understanding the dynamics. So again, this is looking at an individual tuberculosis within a cell, and then starting to understand how that bacteria interacts with the host cell. So here you'll see this is a green labeled Mycobacteria, and it's being surrounded by this compartment in the host cell that will hopefully lead to the degradation of the bacteria later on. And then this is an image of a human lung after it's been infected with tuberculosis, and how it begins to form this kind of ring in the lung, and that's what causes kind of the infection and the sickness that is tuberculosis. And so again, just wanted to highlight, though, that it's not about microscopy. Being a scientist is about looking around you and being inspired to kind of ask questions about what you can see. So again, it ranges from the size of individual slime molds and insects and individual cells to understanding the ecosystems that we live in, the crazy weather that's going on, as well as the galaxy around us. So I love science because I'm able to see all of these different things and observe them with microscopes. And I hope that you guys can kind of appreciate that and learn to love it too. And so again, this is just a little introduction into me, and I hope you guys are kind of interested now in maybe exploring some of these things about science.